We might as well roll, yeah. All right, welcome. Thank you all for coming today. For those of you who were at the lecture last night, David needs no further introduction, but for those of you who are just joining us, David is coming to us from the University of North Carolina at Chapel Hill, where he's been a professor for the past 30 years. So David is a Texan who did his early career stages at the University of Texas, Austin, including majors in zoology and geology when he was an undergraduate, before transitioning to biology and zoology for his MS and PhD. His PhD was done under Jim Bull and Mark Kirkpatrick. And while David was a PhD student, he enjoyed going herping with people like Bob out in the deserts of Texas and the wild areas around there. So I think that resonates probably with a lot of us. So David then did postdocs at Arizona State, University of Arizona, and Cornell. And he did his first professorship at University of Illinois before moving to UNC. So he and I share the same migratory trajectory there. So you can take a biologist out of the desert, but not the desert out of a biologist. So David continues to make annual trips to Portal, Arizona, where his study system of spadefoots is native. So they live in these monsoon pools that he'll tell us more about. So these cross-country road trips often include members of his lab, members of his wife Corinne's lab, and family members. So this has led to tons of productive research. This includes features in UNC's research stories entitled The Frog Family, chronicling their desert adventures together. And these trips also gave their eldest daughter, Corinne, enough material to publish a novel epigenetic inheritance mechanism while she was in high school. So beyond this, David has had an absolutely wonderful career. He's published, I believe, 161 peer-reviewed articles at time of speaking, and 3 books, with another one in prep. And he was recently named a fellow of the American Association for the Advancement of Science. Among these accomplishments, David's a dedicated mentor. He was the Royster Society Distinguished Professor for several years at UNC, which is a program designed to advance graduate studies by offering sort of a society for graduate students. And David has also had many grad students of his own, 16 of them to date. And he also, I think, has had a stellar track record publishing with undergrads. So I think that is something that really marks him as a dedicated mentor. And he also does a lot of outreach. So we heard from him about, like, in a very public-facing way last night. And you can also catch him on BBC and National Geographic from time to time. So without further ado, David, I'll let you take it away.
Thank you, David, and thank you all for coming today. It's a fantastic opportunity for me here. It's really had a blast, all the tide pooling yesterday, and seeing one of my former buddies from graduate school, Bob. And I could tell you all probably a few stories. And one of my favorite former graduate students, David. And so it's a real privilege and really fantastic to be here, as I said. So what I want to talk about today is I want to talk about, when I see an old friend, Dave, there, that I didn't see you last night. I want to talk about the ecological and evolutionary consequences of phenotypic plasticity. And so I'm just starting with a slide of some really spectacular examples of plasticity. So these are all examples of where you get really profound and really pronounced variation within species, within genomes. And you can see some examples from plants and from insects and vertebrates here. But I think most of y'all are already familiar with plasticity, but I just want to start by just defining it. And I'm going to just define it broadly as the ability of an organism to alter its features in response to variation in the environment. And so a lot of things that we think about as maybe not plasticity are actually examples of plasticity. So like condition-dependent mate choice, any sort of environmentally induced differential gene expression and so forth. So many physiological phenomena, as we'll see today, are examples of plasticity as well. And so plasticity is something that I think we still struggle with, especially in evolutionary biology, but it's a phenomenon we've known about for a really long time, right? And so it's nothing really new. And so naturalists have long known that the environment can modify many traits. And this is one of the first sort of, I think, scientifically described examples of plasticity. And this is from Charles Lyell. And maybe you'll know Lyell was a mentor for Charles Darwin. And he's really one of the fathers of geology. And in his big geology book, he talks about this example of plasticity from hydrangea. And so he says some curious experiments recently made on the production of blue instead of red flowers in hydrangea illustrate the immediate effect of certain soils on the colors of petals. So he recognized that the soil type that these flowers or these plants were living in could alter the flower color. And he goes on to say that this variation that you see here, he says, is really similar to what you might see between different species. And so he said this could mislead people into thinking that they're looking at different species when really what's happening here is there's something happening in the environment that's causing this variation within the same species. And sort of picking up on that idea, August Weismann, in the latter part of the 19th century, also studied plasticity, and among other things, he conducted experiments with butterflies and noted that depending upon the temperature and the photoperiod that the caterpillars were reared at, you could get very different butterfly color patterns being produced. And so, he noted that, in fact, the variation here, again, going back to what Lyle talked about, he noted that the variation you see among these different seasonal forms was really as pronounced as that you would see typically between different species. So this suggested to Weismann and to others that plasticity potentially is an important phenomenon for evolution. It might play a role in explaining the origins of species differences. And so he talks about how that might play a role. But these ideas were sort of mostly cast aside, I guess you could say, in the middle of the 20th century with the sort of development of the so-called modern synthesis. And this is a quote from the book, The Modern Synthesis, by Julian Huxley, who's the person who sort of coined that phrase. And he says, evolution results from natural selection acting on genetic variation. Now, most of us wouldn't disagree with this statement, of course, but notice the word here, genetic. And so many people sort of mistakenly assume that when we're talking about plasticity, we're talking about non-genetic change, right? And so even today, many people will argue that, plasticity is something we can ignore in evolution because it's really phenotypic change that doesn't really have a genetic basis. We're going to see the fallacy in that argument here in just a moment. But the point here is that with this emergence of the modern synthesis and, of course, the parallel emergence with the field of genetics, plasticity was largely dismissed. And yet, we know that plasticity is really ubiquitous. And so I would argue that it's really one of the defining features of life, right? So all living things, if you think about definitions of life, and there are lots of definitions out there, and there's some problems with the various definitions, but one of the definitions is that living things are things that can respond to variation in their environment, right? And so all major groups of organisms, from bacteria, which exhibit things like quorum sensing, to plants, which in this case they deploy the roots. Many plants will deploy the roots differentially depending upon the presence of competitors. To fungi, as I said, condition-dependent mate choice, and animals, and even in humans, things like sweating, it's a common form of plasticity, or shivering. So these are all, again, manifestations of plasticity. So this again suggests to me that this is something that we should think about. Now, I was trained and still consider myself an evolutionary biologist, and one of the things that's remarkable to me, until about 10 years ago, you could look at the index of all the major evolutionary textbooks for college students, and plasticity did not appear in the index. I mean, that's changed recently, but this is an example of what I'm talking about, where this phenomenon has been mostly ignored, right? So what I want to do today is I want to sort of look at this problem from a couple of different perspectives. And we're going to first begin by talking about the evolution of plasticity. So as I mentioned, plasticity is ubiquitous. It's only a defining feature of living things. But we're going to see that some organisms evolve higher levels of plasticity, some evolve lower levels of plasticity. What explains that variation? How does plasticity itself evolve? We're then going to look at an ecological consequence of plasticity, and this is just sort of a little vignette because there are many ecological consequences of plasticity we could talk about here today, but one that I'm particularly interested in is the effects of plasticity on facilitating the coexistence of competitors. And so we're going to explore that a little bit. And then I want to talk about two evolutionary consequences of plasticity, of plasticity's evolution, given that it's evolved. So one is innovation and diversification through a phenomenon that we've referred to as plasticity-led evolution. And then I want to focus on how plasticity might really play an important role in basically buying time for populations that face environmental variation in order for adaptive evolution then to occur. And then I want to just say a few words at the end about the implications of plasticity for evolutionary biology. So let's begin by talking about the evolution of plasticity. So the reason we think plasticity is so widespread is because of the following point. So all organisms experience environmental variation. I don't need to show data on this. We all know this, right? We're all ecologists, we're all evolutionary biologists. We recognize this. So natural environments vary. They certainly vary if you look over evolutionary time scales, but many environments also vary within the lifetimes of individual organisms. Now, the reason that's important is because environmental variation decreases fitness. And the idea here is that, selection should favor organisms that produce phenotypes that are a match, roughly, for their environment. And if the environment is changing, then that is going to disrupt that match, basically. And so we can just think about this sort of in a cartoon way. So here we've got this, these insects living on this leaf, let's say, and let's say they blend in well to the background, perhaps to avoid predation, let's imagine. So initially their fitness is high, but let's now imagine over time, and this could be developmental time, of course, it could also be evolutionary time, but let's imagine that now the environment changes and the phenotypes are now staying the same. So now you've got that greater mismatch, and of course now fitness is going to decrease as a consequence of that. In our hypothetical example here, it could just be because now they're more conspicuous to predators. So basically, when organisms experience this environmental variation, of course, we all know they can recover fitness through adaptive evolution. We all learn that in our basic evolutionary courses that we took, right? So you might see the evolution of now new patterns and new colors on these hypothetical insects over evolutionary time to now better match their background. And there are some really good examples of this, right? So this is one of the classic examples, the peppered moth, Betuliaria, where you all know the story, where their background changed, and so you got this melanic form evolving from the more typical form that became rare then. And then with the improvement in, because of the soot covering the trees and killing the lichen, but then in the 1960s and moving forward now, the environment's changed back the other way. So now that melanic form is disfavored and now the typical form being favored. And so we're seeing the recovery of fitness here through adaptive evolution. So this is, again, is a classic example of that, and we know many stories like this that show that. But organisms can also alternatively recover fitness when environments vary through phenotypic plasticity. And so here we're looking at an example of this. These are caterpillars that are changing their colors to match their background. Now I love this example. This is from Mama Nora's lab, a good friend of mine. These are actually peppered moth caterpillars. So here we have this one system. The adults, the adult moths are responding through adaptive evolution. Here we have the larvae, the caterpillars, evolving through plasticity. What's also really cool about this example, by the way, is that you can actually blindfold these little caterpillars and they can still match their background. So it's kind of like they're seeing with their skin, so it's a really neat example of that, right? So, the reason that this works is because plasticity is often adaptive, right? So I talked about this example last night, for those of you who were at the talk last night, but these are water fleas, Daphnia, and as you probably know, these will produce this predator-resistant form when they experience chemical cues from predators. So when they experience these chemical cues from predators, they'll develop this helmet and spine form. And some really nice work by Anil Agrawal and his colleagues have shown that, if you look at survival of the typical and the predator-induced form, they have a much higher fitness in the presence of predators. And you can see the non-induced form or the typical form have much lower survival, right? So this is telling us that this plasticity in this case is adaptive. So, but again, the problem that people will say is that, well, okay, so that's cool, but plasticity, maybe people will argue it's just sort of a non-genetic effect, right? And it has no consequences, further consequences from evolution. But plasticity is often underlain by heritable variation. In fact, in almost every example that I can think of where people have actually investigated this, they have found this sort of underlying heritable variation in whether different genotypes respond to any change in the environment and the manner in which they respond. And so let me just give you an example from my lab. So this is some work that my now former graduate student, Emily Harmon, did, where she's working on these rotifers. And in these rotifers, they have a kind of an interesting example of plasticity where it's transgenerational plasticity, where depending upon what the mother ate, the offspring develop different forms. So if the mother is exposed to vitamin E, usually by eating other animals, then her offspring will produce this sort of like spaceship form that you see here on the right. If she doesn't experience the vitamin E in her diet, then her offspring will look like this sort of typical form on the left there. And so, we're going to get into this story later on, but the main point I want to make here is that these different lines over here, this is basically what's called a reaction norm plot. And so these different colored lines are different clones, different genotypes, and you can see there's a statistically significant what's called a G by E interaction, telling us that there's a genotype by environment interaction. A really simplistic way of thinking about this is that the different lines are crossing, right? So they're not all responding to the same change in the environment similarly, okay? And so basically a reaction norm then is the phenotypes of a single genotype across a range of environments. But the interpretation of this, when you see this kind of plot, is that that's telling you there's underlying genetic variation, right? And I should be more proper and say it's underlying heritable variation, because we couldn't rule out that some of this could be epigenetically mediated. And that's the really key point, is that there's heritable variation here, possibly genetic variation. Now, that's important because we've already seen now that plasticity can impact fitness, and now we're seeing that plasticity exhibits heritable variation. So now we have the three conditions that need to be satisfied for adaptive evolution to occur, right? Variation, differential success, and heritability. And so we then expect it to undergo adaptive evolution. And so these are examples of just some part of a working group that I was on a number of years ago, where we looked at, led by Courtney Murren, where we looked at evidence for evolutionary change and reaction norms. These are continuous reaction norms. And there's lots of evidence that all kinds of organisms, plants, animals, invertebrates, vertebrates, do indeed evolve different reaction norms, right? And so we're looking at this in the three different ways. So in terms of the offset, in terms of the intercept, if you will, in terms of the slope, but also in terms of the curvature, right? So there's all kinds of possibilities. In other words, reaction norms can evolve in all these different ways. So why does plasticity then evolve? What are the conditions that will favor its adaptive evolution? Well, typically, we tend to find plasticity evolving to greater values or more enhanced plasticity when organisms experience variable environments, right? And so this is one of the early studies showing this, and this is really nice because it's from natural populations. These are can't remember what frog species this was, but it's the common frog, I think, from Europe. And in this case, the researchers were looking at these frogs from different populations on islands off of Sweden, and they were looking at a measure of environmental heterogeneity that these different populations experience. In this case, is the coefficient of variation in pond drying. So some ponds dry faster, some dry more slowly, but there's also, of course, variation in that. And then they looked at plasticity and developmental rate. So frog tadpoles and many species, they have the ability to facultatively speed up their development if they sense that their pond is drying up, right? And so these researchers measured, again, environmental heterogeneity, and then they had a measure of plasticity. And as you can see, there's a significant positive relationship here, right? So telling us that indeed plasticity is enhanced in populations that are experiencing higher variation. And many people have done these kinds of, using experimental evolution studies, shown that you can evolve plasticity in the lab in organisms when they experience more variable conditions, right? So that's how you get the evolution of increased plasticity, but plasticity can also go the other way, right? So you could also lose plasticity and you get the evolution of decreased plasticity. And this tends to be favored in constant environments. It's a little bit more to the story than that, but just think about it as constant environments. And this is again an example of this possibly happening in natural populations. And this involves tiger snakes. And if you don't know, tiger snakes don't do this at home, right? So tiger snakes are super venomous, right? So they're not a fun model organism to work with. But in this case, the researchers were looking at tiger snakes from the mainland of Australia, but also various islands off of Australia. And on the picture up there, what you see is on the left, these are tiger snakes from the mainland populations, and they tend to be born with small heads, and then they will develop large heads facultatively through plasticity when they eat large prey. The snakes over here on the right are from islands off of Australia, and they're born with larger heads because these islands presumably don't have a small prey. But what the research has shown, again, the data, there's not a lot of data here, but it's kind of interesting. They're able to calculate the isolation time, how long these different populations of these islands have been isolated from the mainland population. And then they again had a measure of plasticity here. This is jaw plasticity. And what they show is the longer they've been isolated from the mainland, the less jaw plasticity they possess. So in other words, they're losing plasticity that presumably they once had because these are derived from the mainland populations. They're losing that plasticity as they experience more constant environments. So this is sort of the idea that, again, this is sort of a controversial idea in the plasticity world, but this is evidence for something that Conrad Waddington dubbed genetic assimilation. And so Waddington showed this process where you could get the evolutionary loss of plasticity as well as selection. He showed it with his experiments in fruit flies, where in this case, in one experiment, he was selecting on this so-called cross-veinless phenotypes. They lack that vein right there, right? And so we don't know why that cross-veinless phenotype or why that cross-veinless form is adaptive, other than he basically selected against that form, right? So he's basically exerting the selection here. And what he showed by basically exposing them, in this case, to heat, shock, but in other experiments, he exposed them to ether. He showed that over time, he could selectively favor what was initially an induced form, induced through these changes in the environment, where basically it became, as he said, assimilated or incorporated into the genome, where they basically lost all that plasticity. So again, many experiments have shown genetic assimilation in the lab, and we can only infer it from the wild, like in the case of the tiger snakes that I showed just a moment ago. And we're going to come back to this a little bit here later in the talk. So that's a really quick overview then of how you get the evolution of plasticity. So why plasticity is favored, how you get more enhanced plasticity, how you get the loss of plasticity. So I want to just highlight now what I think is an important ecological consequence of plasticity's evolution, and that's the coexistence of competitors. And so we just saw a moment ago with these Daphne examples, how plasticity can play an important role in mediating ecological interactions, in this case, between predators and their prey, right? And so we see that, plasticity allows these Daphnia to be able to, deal with these predators by producing this form that's more predator resistant. But plasticity, we think, is also important in mediating interactions among other type of enemy, and that's competitors. And this is an idea that's been around for a long time, and it's the idea that plasticity can basically help organisms mediate niche partitioning or resource partitioning. I'm just showing this with this cartoon off the web. But basically, you've got, like, imagine some birds that are utilizing both the trunk and the crown of this tree and even the ground, to get their food. And then let's imagine another species moves in. The red birds now here move in, and they start utilizing the trunk here. And so they basically, through this sort of resource partitioning, our initial yellow species then shifts up to feeding in the crown, whereas the red species shifts to feeding on the trunk here, right? And so there are many examples of this sort of resource partitioning from various groups of organisms, and some of y'all may know more about that than I do, right? So we don't need to really burrow into that too much. But there's also now some recent evidence then that this sort of plasticity-mediated resource partitioning can play an important role in enabling species to coexist. And so you would imagine that if you have some mechanism whereby organisms can basically, move into utilize a new resource, that should minimize competition between them. And some really nice work that's been done over the last few years by Mark Turcott and Jonathan Levine and their collaborators has indeed shown both theoretically, but also they've done some really nice empirical work showing that this does seem to work this way. That plasticity does enable potential competitors to coexist by basically partitioning out different resources or different niches more generally. So the question then becomes, well, what are the longer term consequences of this? Because does this lead to any evolutionary consequences? And so this sort of plasticity mediated resource partitioning, the question is, can it then promote adaptive evolution and really allow those competitors to coexist on a longer term basis? And one of the things that I sort of started working on this question a number of years ago, and after I'd sort of been working on it for a while, I ran across this quote by E.O. Wilson in his book, The Diversity of Life, where he's basically describing this process whereby you get resource partitioning and then you get genetic assimilation of those differences. And so what Wilson says here is he says, imagine a case in which two species have been squeezed together in the same communities long enough for evolution to occur. When they first came into contact, they were elastic and could diverge in their habits enough to lessen competition. So in other words, he's talking about they were able to shift onto different resources using plasticity. And then he says, as the generations passed, genetic differences arose and hardened the distinction between the two species. They experienced the evolutionary change called character displacement. Now, Wilson is relevant here because Wilson, along with another scientist, is the person who coined this process, character displacement. I actually think it's a horrible phrase. I think it sounds like, I just don't like the phrase, but this is what we call it, right? And so really what he's talking about here is how character displacement could come about through sort of initially through plasticity, mediating shifts through competitors. And so the question is, does this actually happen in the wild, right? And so one reason why we expect plasticity to really play an important role in facilitating character displacement is we can kind of think of a cartoon here where let's imagine we have a non-plastic population that experienced a competitor, and let's look at now a plastic population. And so if we have a non-plastic population, and let's imagine another, this is number of individuals over here, this is a phenotype under selection down here, but let's imagine this is a phenotype involved in resource use, in our case here. And let's imagine now a competitor invades the environment of that initial focal species, right? Now, if that species doesn't have plasticity, the only way it's going to really be able to move away from that competitor is to, evolve new variants, right? Now, those can evolve through mutation, but they could also come in through, introgression or gene flow, potentially recombination, things like that. But in every case, though, these processes might unfold fairly slowly, right? And so it may take a while for the character displacement to occur. And the reason time is important here is because character displacement is kind of a time-limited process, we think, right? So if it unfolds too slowly, the alternative outcome of competitive exclusion might transpire instead of character displacement. Now you contrast that to a case where you have plasticity. We're looking here at an extreme example of plasticity where you have different morphs, but it would also work you have more continuous variation here. Now we have the competitor invade, again, the same environment with our focal species, but basically you have this plasticity now. You can basically shift away from that competitor by just sort of differentially, the relative frequency of those two different phenotypes. In this case, again, simplistically, we're looking at two phenotypes, but it would also work with continuous variation, where they basically sort out that pre-existing variation. So here we think character displacement should transpire much more quickly. So going back to Wilson's quote, he's suggesting a scenario, and we think that scenario is probably going to be probably pretty common and pretty important in the wild because of the fact that plasticity should facilitate character displacement. So we've been studying this process in the spadefoots, like David mentioned before. And so these are the spadefoots that are found in North America. There's some old world spadefoots too, but these are the ones we have in the New World. We recognize at least 7 species here in North America. We think there's probably more diversity represented here. Of course, you actually have 2 species here in Oregon. You have Intermontana. Actually, this species, Hammondii, I noticed in the collection last night, there is actually Hammondii from the southern part of Oregon here, so that I need to correct that range map. But one of the things you see here is that you see that their sort of greatest diversity is found down here in the Southwest. And so as David said, that's where I've been working for a number of decades now. And so I want to just talk to you a little bit the natural history of these interesting creatures. So they're called spadefoots because they've got this keratinized spade on the back of their feet, and they use that spade to dig because this is the kind of typical habitat in our study sites in southeast Arizona. And so this is not a really great place for amphibians to live. And so the way they're able to live in this environment is by basically burrowing underground, and they just stay there for most of the year, right? But they're amphibians, and so that means they have to have water in which to breed. And in the desert Southwest, and probably here in Oregon too, you get these sort of rainstorms, in this case, summer rainstorms. And so that's really what sort of starts the process off. And so these rainstorms cause water forming these low-lying areas and these little temporary ponds. And it also causes the adults, and I think I need to turn on my sound here. Hopefully this works. So this is just these are two male Mexican spadefoots. I wish I had a better way of showing you this, but you go out in these ponds, and the ponds could be, on the larger end, they might be the size of this room. On the smaller end, they might be the size of your bathtub, right? And so, it rains, and all of a sudden you see all these frogs appearing out of nowhere, right? And they congregate in these ponds and start calling. And all the mating happens in a single night. So if you miss that one night, you're screwed until next year, or maybe the year after that, or whatever. And then the tadpoles hatch really quickly, and then they're basically in a race to get out of these ponds before they dry up, because these are ephemeral pools in really arid environments, hot environments. And so in a lot of years, I mean, like a couple of years ago, we didn't see anybody get out. So all the ponds dried up before any of the tadpoles metamorphosed. But here you see this little guy moving on the land. So he's got his four legs, and these other blobs back here are other tadpoles in this pond that's drying up. And so they may or may not make it out of it before it goes completely dry. They actually have other tricks up their sleeve, by the way. So one year I was taking a picture of what I thought were just tadpoles desiccating in this, almost completely dry mud. And I kept hearing this popping sound, because I was focusing. It's like, what is that sound? And finally I just focused in on one, and his mouth, and so they were still alive, encased in the mud, just barely moist mud, just like presumably waiting for another rainstorm to fill the pond. One of the things these guys do that other amphibians don't do as larvae is they develop lungs really early, presumably because of what can happen where the pond dries up, and then they may need to live for a while waiting for another rainstorm to potentially fill up their pond again. And sometimes it happens, sometimes it doesn't, right? But anyway, so the thing that, and some of y'all may have seen this video last night, so one of the things that I'm fascinated with this system is that there's this really extreme form of plasticity present. So you see a lot of the tadpoles look like these guys around the margin, and then you see within the same pond these large, kind of odd-shaped individuals, and if you watch them, they're like predatory. So they're kind of like little sharks just swimming around in these little pools, and you'll every now and then watch them nab one of the other ones and carry them in the deep as they scream and whatever, just kidding. But, so they eat other tadpoles too, right? And so you have these two different morphs, which we call the omnivore morph and the carnivore morph, because they basically occupy different trophic niches. And by the way, the two tadpoles you see here, they're the same age, they're from the same pond, they could be from the same clutch. So this is the example of the amazing variation you get in this system. And so one of the things I'm not going to show you today, but you can sort of quantify the shape variation here. And the shape variation is much greater than you see between different species of spadefoots, and actually between, I don't know if you noticed the earlier slide, there are two different genera of spadefoots here. So it's actually much greater, going back to what Weissman and Lyle talked about. You're seeing here an example where plasticity is generating variation, which is really in the order you see between different genera, right? So, anyway, so we got these two morphs. They differ. Importantly, they differ in their digestive morphology and the digestive physiology. We're going to come back to this later on, but this is the omnivore's gut. You're basically looking at the bottom of the tadpole because the skin's clear, and you can see if for a little tiny tadpole like this, it might have a gut that's a meter long. So it's a really long coiled gut in there. And then probably wouldn't surprise you to hear that the carnivore has this relatively short gut, right? So it may be a fifth the total length of the omnivore gut. And I'm highlighting that because later in the talk, we're going to look at gut length in response to different environmental conditions, right? But again, they occupy different trophic niches, as their name implies, and you've got basically a detritivore or an herbivore, and then you've got a carnivore over here. Now, we are dealing here with plasticity, and specifically it's diet-induced plasticity. So You can ask if a young tadpole eats live prey, it always develops into an omnivore. Actually, we're going to come back later on. There are some cases where they will still develop into carnivore. But then if they do eat the live prey, they will sometimes develop into carnivore. And I say sometimes because, again, there's underlying heritable variation among different SIB-ships and the whether they respond to that cue, right? So some SIB-ships are really responsive and they're much more likely to produce carnivores. Some never do it. They're just completely unresponsive, right? And so one question we want to ask here before we delve into some of the testing, the idea that Wilson laid out about so-called plasticity-related character space. And I want to just highlight, first of all, why do we have plasticity in this system? Because this is not a typical thing that you see in most neuron tadpoles, developing these distinctive morphs like this. And we think that we have these plasticity in this system because they're experiencing really variable conditions in their environment. And so just to show you an example of this, these are pictures of the same pond in consecutive years. And so this is showing you the kind of year-to-year variation in volume of the pond as well as in the resources in that pond. So here we are in 1 year where this pond just barely fills and it's got their tadpoles in it. And then here we are in the very next year, and that's the same spot that I'm taking the picture from, and you can see the pond's much larger, right? So this situation, obviously, the pond's going to last a lot longer than over here. This one's going to dry up a lot faster. But the other thing that's going to be different between these two ponds is the kind of resources in them, right? So here you're going to have, all the fairy shrimp that these guys feed on, but also other tadpoles. They're going to be really concentrated in here, right? So there's going to be a lot of meat here to eat, right? Because it's really, really highly concentrated. Over here, the meat, the shrimp, and the tadpoles are going to be really diluted. So a meat eater is going to have a harder time over here. But also because it's really wet year and all the stuff getting washed in, you're going to have a lot more plant material and detritus building up over here as well. Now, the reason that's relevant is because, these are relatively long-lived animals. They can live 15, 20 years in the wild. So any given female, 1 year she might face a situation where her offspring might face a situation where they're going to be in a pond that's going to dry up really fast. The next year, it might be a situation where the pond's going to dry up more slowly, right? So you get this just really profound variation here. So why does that matter for the tadpoles? Well, the carnivores appear to be favored in rapidly drying ponds because the carnivores, because they get the more nutritious food source, they develop faster. And so they're much more likely to make out of these ponds. They can reach metamorphosis in about 2 weeks, depending on the species, whereas the omnivores might take 3 or 4 weeks. And that week or two might make a big difference. So here you see these omnivores desiccating, and you see this little carnivore moving on the land. And so not only are they more likely to make it out, but they also are bigger at metamorphosis. So this is looking at size at metamorphosis of carnivores and omnivores from the same pond. And size is a good proxy for fitness in the system because bigger is typically better. They survive better in the post-metamorphic phase. They also then will tend to be bigger adults, and then males have higher mating success, females higher fecundity. And so you can see these carnivores are being favored in these rapidly drying ponds for both survival, but also just in terms of their body size. But now you look in those slow drying ponds, and now you see the advantage goes in the opposite direction. So now the omnivores, they're both able to get out because the pond's going to last long enough. But when they get out, now the omnivores catch up to the carnivores in body size and they actually come out larger. And so here you can see an omnivore. It's just basically like a golf ball with a tail on it. It's this fat little guy. And here's a carnivore, and he's just shrinking away because what's happened here is that this is a pond where it was initially really shallow. And so some of the tadpoles started developing into carnivores because they could get out of that pond really fast. And then another rainstorm came on and refilled that pond. And so now it's like all the feed is really dilute, right? And so this guy is just starving because remember, he has that really short gut. He can't assimilate, or she, I don't know, but it can't assimilate the plant material in the low nutritive quality detritus because it just has that really short gut. And there are other features that make it not really good at eating detritus and stuff like that. So what's happened here, as I said, is that you also see that not only the shrimp really dilute, but the shrimp that they basically depend on really start declining in density as well because the shrimp senesce really rapidly. So even when the pond's lasting a long time, the shrimp basically are there for maybe a couple of weeks and they're gone, right? So there's basically no food for them. So again, we think we have this plasticity in this system because you get a variable environment and different phenotypes are favored in different environmental conditions. But also importantly, and I'm not going to talk about this too much today, but the environment is also predictably variable. So they actually have a way of assessing whether they're in a fast drying pond or a slow drying pond. And we could talk about that later on if you're interested in that. So let's go back to this idea. That's a really long overview of the study system. Let's go back to this idea about um, using spadefoots to test this idea that Wilson first laid out about so-called plasticity-led character displacement. And so it appears that spadefoots have indeed undergone this process. And so here we're looking at two different species, Spia bomifrons, which is basically found in the Great Plains and in New Mexico, and the Spia multiplicata, which is found down in Mexico and then also found in New Mexico, right? So you've got each species occurring in allopatry, that is by itself, and then you've got the two species potentially occurring here in sympatry, this zone of overlap in New Mexico and surrounding states. And so if you look at what's happened here, if you look at the color codes here that match the map over here, so we're looking at the proportion of carnivores that are produced by each of these two species when they're both in allopatry versus when they're in sympatry. And what you see here is that in allopatry, so the multiplicata in yellow, the bomifrons in blue, they're producing about equal frequencies, intermediate frequencies of carnivores, right? But now when you go here in sympatry and shown both cases in the green, you can see that the one species start to produce almost all omnivores, very few carnivores, and then the other species is the opposite. It's producing almost all carnivores, very few omnivores. And so what's interesting is that you can also do an experiment in the lab where you can basically simulate first contact between these two species by now taking animals from allopatry of each of these species. So the two yellows are the multiplicata from allopatry, the blues are the bomifrons from allopatry, and you can rear them in pairs, but sometimes you rear them with a conspecific, but other times you rear them with a single heterospecific. And so we're simulating the first contact between those two species. And what you can find that's really cool is that they basically sort out these two different ecological niches through just plasticity. So what's happened here is that, again, this species is producing almost all omnivores. This species is producing almost all carnivores. And we can get into why I think the proximate mechanisms for that are, but basically it's just this one's a little bit better at eating the shrimp, which is one of the important cues that induces the carnivore. You know, we don't know why they pre-exist with that advantage or not, but that's something we're looking into. But the important point here is that these plasticity-induced niche shifts, shown here in the bottom graph, they mirror the niche shifts, the sort of fixed niche shifts, which we see in the wild. And we know these are fixed because we can bring these in the lab, and that's what they do, right? So this implies, again, if we imagine that this is how evolution happened in this system. And I should have mentioned sympatry is secondary contact here. So we know the ancestral condition is allopatry. And the secondary contact is occurring here because the one species, bomifrons, is actually expanding its range out of the Great Plains southward and westward into the deserts. And again, there's an interesting story there that we could talk about. But the point here is that it looks, if we infer this is what they were doing when they first came into contact, and then those differences hardened over time, again, as Wilson suggested. So it looks like there's evidence for plasticity-led character displacement. But there's one other twist to the story that I think is kind of interesting. And that is that if you look at the species that has more recently encountered the competitor, and so we're going to look at what's happening in multiplicata. And multiplicata in southeastern Arizona has only recently experienced competition from bomifrons, because remember, bomifrons is coming down out of the Great Plains, right? It's moving southward and westward. So when we're out there in southeastern Arizona, we think that we can't obviously time this, but we think it's evolutionarily much more recent for multiplicata. And if we look at what's happening with multiplicata, what we find is that it looks like the character space in multiplicata is mediated by a maternal effect, right? So it's kind of a non-genetic inheritance, like what we were talking about again, for those of you at the seminar last night. So if we look at what's happening in allopathic populations of multiplicata, it's just what we showed earlier. You get these two different morphs. They, on average, you get relatively large females, and they produce relatively large eggs, and then they produce omnivores and carnivores in the next generation. But when you look at what's happening in sympatry, and these ones that recently contacted the competitor, remember, they're only producing omnivores. I mean, there are a few carnivores being produced, but almost all omnivores are being produced. And so the result of this is these omnivores are smaller, so they tend to mature as adults as smaller, poorer conditioned adults. They're just not as fat for body size, too. Now, those females, for reasons we don't totally understand, but they tend to produce smaller eggs, right? So they're producing smaller eggs than the females in nearby allopatry. We could do this, by the way, because there's an elevational gradient. And we can compare populations that are just a few kilometers apart, one that is experiencing competition with the other species, with bomb fronds, and one which is not, when you get higher elevation, right? Now, those smaller eggs then are more likely to develop into omnivores when they hatch out. So we think that when they first came into contact, this sort of maternal effect was the initial mechanism that reinforced producing only omnivores in Multiplicata. And presumably if this persists, right, if this selection persists, then that maternal effect, which is a type of sort of non-genetic inheritance, might get taken over by sort of more permanent genetic changes. We don't know that, obviously, it may not go that way, but it looks like that's what's happening in the other species in bomifrons. It looks like it's more permanent changes. There's not a maternal effect there. All right, so again, this maternal effect of form of transversal plasticity promotes that character splicing. So that's a really quick overview of that story. So that's what we think of an important consequence of plasticity, enabling species to basically move away from enemies, in the case of competitors, move away from competitors, and that could even have potentially long-term evolutionary implications of allowing species to coexist, right? So I want to turn now to two evolutionary consequences. Innovation and plasticity, innovation, diversification versus plasticity-led evolution is the first one I'm going to talk about here. So, there's a lot of debate about, does plasticity have an effect on evolution? And if so, does it tend to speed up evolution? Does it tend to slow down evolution, and so forth? And there are arguments for why plasticity might impede evolution. I'm not going to really talk about those today because I think those are, there's some interesting stories there, but I think those are somewhat special cases. At the other extreme, there are some tantalizing evidence suggesting that plasticity can also facilitate evolution. And this is sort of one line of evidence. This is from stuff from my lab. This is a former undergrad where we did this work. And basically, we were looking at whether lineages, using what's called a replicated sister group comparison, whether lineages that have evolved conspicuous plasticity, in this case, these are resource-based plasticity, like resource polyphenisms, like what we see in the spadefoots, are they more species-rich than their sister clade that has not evolved that plasticity? And in all 5 cases, we find that the more plastic clade is more species-rich. So you see, again, this doesn't mean a whole lot, but you see evidence like this suggesting there's a relationship between plasticity on the one hand and rates of diversification on the other hand. So one of the things we've been developing in the lab is an idea known as plasticity-led evolution. This is basically taking ideas that other people have talked about for decades and just sort of making it a little bit more amenable to sort of how we might think about this in sort of modern evolutionary context. And the idea is that plasticity-led evolution occurs when a change in the environment triggers an environmentally induced phenotype, and then that phenotype is subsequently refined by selection. And so this is really a situation where the new phenotype first appears following a change in the environment. So this is in contrast to what we may think of as sort of the normal way we think of novelty arising through what we might call just mutation-led evolution, where it's some new genetic mutation comes into the population, either through a mutation event or through, it could be through, like I said, integration, like we talked about earlier, or gene flow. And so that's really the contrast we're making here, but we're saying that's really something happening in the environment. And so some of y'all saw this last night. I'm going to just go through this kind of quickly. So this is how we think this process works, right? So we start off with a genetically diverse population, and I'm showing these tadpoles here as all being the same size and shape, meaning they all have the same phenotype. The different colors are meant to signify different genotypes. Now, when their environment changes, they start producing these new phenotypes through plasticity because that's a stressful event. And the way most organisms can respond to environmental changes, as we talked about earlier in the talk today, is through plasticity. But the important point here is that these different genotypes now are responding to the same change in the environment differently, right? So the blue genotype becomes this fat-bodied tadpole. The maroon becomes a skinny tadpole. The green is kind of intermediate between those two. And the orange isn't responding at all, right, in this case. So this is that cryptic genetic variation that's being exposed to selection. This is kind of like what we talked about earlier with the rotifers, right? So when they experience a change in the environment, you now get all this underlying genetic variation being revealed with selection. So once that happens, then selection can start to act on those phenotypes and thereby act on the underlying genotypes. So remember, selection can only see phenotypes. It can't see the genotypes, right? At least not directly. And so let's imagine this fat-bodied form is now the adaptive form for this dark-shaded new environment. So selection can start to refine it and start to produce a larger tadpole here. And eventually you could end up with a novel polyphenism like what we have in the spadefoots, where you get two different morphs being produced in response to different environments, or you might end up with genetic assimilation in that novel form. So in other words, you might get the fixation of that fat-bodied tadpole in this case. But again, the important point here is that this whole process was started off by the plasticity, and that's why we call this plasticity-led evolution. It's because the plasticity really got this process rolling by exposing that cryptic variation to selection. So we've been testing for this idea in my lab, and I've just highlighted 3 former graduate students who are really key in this, who are all now professors themselves at other institutions. And we were specifically interested in knowing whether or not this distinctive carnomorph evolved through this process of plasticity-led evolution. And so the way we tested this idea was to look at these 2 different predictions. One was pre-existing plasticity. One prediction is that pre-existing plasticity must have been present in some ancestral lineage because that got that process rolling. And then secondly, that pre-existing plasticity underwent adaptive evolution in more derived lineages. So we did this in a comparative framework, and so we start off by just building a family tree of the spadefoots here. And so our outgroup here are some various old world frogs, and these are our new world spadefoots up here. And what I'm showing you there, those letters, the O's and the B's and the C's, refer to the tadpole phenotypes they produce, right? So the O means that they only produce omnivores. The B means they have that polyphenism. They produce both carnivores and omnivores. And then there's this one lineage here, I'm going to get to in a minute, where you see the C, that means they produce carnivores. I'm going to say mostly because they do produce a few omnivores. And then the colored, the blue ones, are the observed phenotypes that the larvae produce. And then the gray is the inferred. So we can infer that based on their position on this phylogeny, right? And so what you see here is that the ancestral condition we infer was to produce only omnivores, because that's basically the condition that almost all frogs produce, right, including their sister group here, right? So we inferred that ancestor must have been an omnivore producer. So we really want to study that ancestor, or more specifically, we really want to study this ancestor right there, which is the ancestor to these 2 groups, the modern Scaphiopus, which don't produce carnivores, these are spadefoots, and then this clade right here, the Spia, which do produce those carnivores, right? That's the ancestor we really want to study, but of course it's extinct, right? So we have to use this extant lineage right here, this extant group, as our ancestral condition, right? So we're going to infer, we're going to use this sort of ancestral character state reconstruction. So we've got this ancestral condition up here in blue. We've got these ones in orange that represent the derived condition, and then I'm just going to call this red one our most derived condition, because it's producing mostly carnivores, which is the most distinct character state here, right? So what we're going to do is we're going to take these different groups and we're going to test those 2 predictions that I laid out earlier, right? We're going to basically have them experience different environments. So I'm going to just sort of summarize the results here for sake of time. So we start with the Scaphiopus species. And we do an experiment where we rear some animals on plants, and we rear other animals of the same sibships on shrimp. And then we look at their gut lengths, and I'm just going to summarize those with these red lines here. And then we also look at their performance. So how well they survive, how well they grow on those different diets. And what we find is that when we do that with Scaphiopus, they're not producing carnivores, even in the lab, but some of them start producing slightly short guts in response to eating the shrimp. Some of them actually, I don't know if you can see this here, some of them actually started producing longer guts in response to shrimp. So that's a really important point here, is that the ancestral plasticity is not necessarily adaptive. It's just, there's just variation being revealed, right? So some of them are doing the right thing by producing shorter guts, but a few sibships are actually doing the wrong thing by producing a longer gut, and eating meat. But then you look at the performance, and they actually do really, really poorly on the shrimp. A lot of them are actually dying, and they don't grow very well on it at all because it's a novel diet, and they just don't have the ability to assimilate that food source, right? And so that's kind of what we would expect. Now we look at the Spia species, and they have the polyphenism, and we see this enhanced plasticity. So again, just sort of a cartoon here, but they tend to produce really long guts when they're fed on plants. They tend to produce really short guts. Again, siblings being reared on different diets. And then in terms of performance, they've recovered some of that fitness because they actually do equally well on both diets, right? So both on plants and on shrimp. And then lastly, if we look at bomifrons from the sympatric, and what the sympatric refers to is these are the bomifrons that are in sympatric with multiplicator. So these are the ones where selection is favored producing only carnivores as a result of that as a manifestation of that character displacement. So here, if you feed them on plants, they basically are producing a short gut regardless, whether they feed on plants or on shrimp. So we're starting to see signs of what we think is genetic assimilation. And the other thing we see is they actually, it's the opposite over here. They grow and survive really well on shrimp, but they don't do so well on plants. And again, that makes sense because they're producing this really short gut. And so we infer that what has happened here is we're looking at, different snapshots in evolutionary time. And so we infer from this that we are indeed seeing the evolution of this novel complex phenotype through this process of plasticity-led evolution. Now, I just want to highlight that this is not the only example in which we have good evidence for plasticity-led evolution. We also have from cyanobacteria. Some have argued that cast and social insects, there's good evidence that's evolved through that process. Again, with a former undergrad, we actually suggested the rattle and rattlesnakes evolved through this process. And even to show you it's not just the origin of complex, it could also be the loss of complex phenotypes. There's some really good evidence that eye loss in cavefish has also occurred through this process of plasticity-led evolution. So again, there's increasing evidence for this idea. So I want to just talk about another way that plasticity can in fact impact evolution. And in my way of thinking, this is probably the most common way that plasticity affects evolution. That's by buying time for evolution to occur. And the hypothesis is as follows. The hypothesis is that, because plasticity is often, not always, because it's often adaptive, that can enhance population persistence and thereby buy time until populations can acquire more permanent genetic changes that can adapt to locally rapidly changing environments. And so we've been testing this idea in this rotifer system that I described earlier, and this is Emily, who's my former graduate student, now postdoc at Duke, and has this really cool example of plasticity where these rotifers will develop into these different forms depending upon the maternal diet. They also have plasticity in their sexual mode, so they will produce either asexual or sexually, and there's a, depending upon conditions as well. And so what she did was she was able to do an experimental evolution study where she took these different clones of these rotifers that had exhibited different levels of plasticity. So, different lines, some were more responsive to the vitamin E diet, some were less responsive. And then she exposed them to 24 replicated environments. And then basically, because they have really short generation time, she could look then at the persistence of those different lines. And basically what she found is that populations of clones that had higher levels of plasticity, they actually were the ones that persisted longer in her replicated environments. So again, showing us that plasticity indeed can allow persistence. But what about the evolution part? Well, she also showed that populations of these clones that express higher levels of plasticity also evolved more, right? So this is telling us that indeed plasticity can not only explain persistence, but it can also explain why some lineages might evolve faster or be more likely to evolve because they've basically been able to buy that time. And this, I think, is probably the most important effect that plasticity has on living things. It just basically allows them to stay in the evolutionary game long enough for these changes to sort of accumulate. So I want to just end by talking about some broader implications here. So, we highlighted at the beginning how this, the modern synthesis sort of developed and it basically this statement here from Julian Huxley is that, when he says its evolution results from natural selection acting on genetic variation. Obviously, he's leaving out other evolutionary mechanisms, right? And he's focusing specifically on genetic variation as opposed to other heritable mechanisms like epigenetic effects and so forth. Now, there's been a debate, and some of you are probably aware of this debate. There's been a debate sort of going on for 10 or so years now where people have been arguing that maybe we need to extend the evolutionary synthesis or maybe we need a new evolutionary synthesis because it's not incorporating phenomena like plasticity, but it's also not incorporating phenomena like non-genetic inheritance and some other things like niche construction and so forth. And, these are all good scientists and they've been doing, they're friends of mine and stuff. I have no problems right here. So new books coming out and so forth. And so, every now and then I get sort of asked, well, where do you fall in this debate? And I just say, I'm not into this stuff, right? And let me just end by just telling you, the ideas we've looked at today, the buying time idea and the plasticity-led evolution idea, there's nothing radical here, right? So I can't stand up and say, hey, I got this new idea, right? This is basically what Darwin talked about, right? These both entail selection acting on heritable phenotypic variation that affects fitness, and that's the standard model of Darwinian evolution, right? That's the standard model of adaptive evolution that we've had since Darwin. And of course, Darwin didn't know anything about genes and so forth. So it's an idea that's really stood the test of time. Now, I'm not talking about some of the other phenomena that these colleagues are also talking about, like niche construction and non-genetic inheritance. Those do get a little more radical, right? But we're talking about plasticity here. There's really, it fits very comfortably within, I think, the so-called modern synthesis, whatever the heck that is, right? But I do want to end by saying, incorporating plasticity more fully into evolutionary biology, I think is really essential. So, I mentioned at the beginning, it kind of bugged me, that plasticity was never even mentioned in indices of textbooks. And I think the reason why incorporating it more fully in evolutionary biology is important is because it can emphasize how the environment can both act as a selective agent, but the environment can also help determine the origin of those phenotypes in the first place. And that's something we often forget, right? So if you think about genetically encoded traits, right? You think about just some magical process whereby a gene or set of genes produces a trait, and you forget that that trait had to be produced through development, through the interaction of that genome with the environment. So this plasticity is really critical in how phenotypes come about in the first place, which selection then can act on. But the other important point here is that we've seen that plasticity itself can evolve. It's not just something that's sitting out there that's non-genetic, as people will say. Plasticity is like any other trait, as long as it exhibits those three qualities, that we talked about earlier, variation, differential, replication success, if you think about broadly, and inheritance, it can evolve by selection, right? So plasticity itself can evolve. And furthermore, that evolution of plasticity, as we saw today, can in fact facilitate further adaptive evolution. So we looked at this idea of plasticity-led evolution. That may, we think, play an important role, for example, in character displacement. Without that plasticity, we're not sure that character displacement would happen, right? So I want to just end by thanking you and thanking some of the people who played a role in this work. These are some of my former graduate students, now some of the funding agencies. And I know you see there's not a former member of my lab on here, because he didn't want to work on spadefoots for some reason. But I want to assure you that this missing person is on my Mount Rushmore of former students. So there's David looking very, very stoic. So, and again, thank you very much for the invitation.
Right, so I think we have time for one or two questions here, so I can field the mic, and anyone who wants to ask, just go ahead, jump in. Yeah. All right, I think I saw Sylvia's hand first.
Don't worry, I can project.
I think we need for the people on Zoom, maybe, right?
So I saw, is that on?
Should be. It's fine.
Okay, so I noticed that you had a lot of examples of plasticity in smaller organisms and amphibians and reptiles, but not a lot and from last 90 birds. But do you know of any examples of plasticity in mammals? Is it just not as common in this group?
So there's enormous amounts of plasticity in mammals. So mammals show a lot of behavioral plasticity, right? So that's all plasticity. Mammals show, like all other organisms, also show a lot of, physiological plasticity. So like at the transcriptome level. So say to take breakfast, I mean, there are different genes being expressed now in my body than after this big breakfast than there was before, right? And so, mammals exhibit a lot of that plasticity. Now, people used to say that plants were different from animals, especially like mammals, because they said, well, plants show a lot more plasticity than animals. And the reason, of course, is that plants don't. I mean, I happen to think plants kind of behave actually, but some people say they don't. And so people were looking strictly at morphological plasticity. So when you think about morphological plasticity, mammals don't show a lot of that, although we do show some morphological plasticity, right? So go work out on weights for a while, you'll big muscles, and so forth and so on. But yes, plasticity is definitely present in mammals. Aren't
saw a couple other hands next. Like Mark had a question.
So I had a question thinking about plasticity. This is optimal gift persistence, right? How do you think that interacts with sort of adaptation from recombination of standing genetic variety? It could be happening on a similar time scale. But I agree that those are some.
So basically, good question. So basically, the plasticity-led argument, plasticity-led evolutionary argument is basically evolution from standing variation, right? So that's, I want to be clear on that, right? And I mean, as you alluded to there, I think often they're both occurring, right? So these are not mutually exclusive. And I also want to be really clear, it's sort of, they're not mutually exclusive with mutation-led evolution either. And so we're not, in any one system, you're only going to see one process working. What because I couldn't get into all the details here, but the carnivore morph is a complex phenotype, like I said. There is a specific trait in that carnivore morph that they produce. We call it a palate spike. It's a new trait we discovered when we were studying this. We don't think it's sensitive to the environment. So we think that component of being a carnivore might have evolved through mutation-led evolution. We're pursuing that more in detail. So even within that one complex phenotype, we think those two processes can be working at the same time. And certainly the buying time process can work. If you want to think about it that way, also going back to the maternal effect, acting as a bridge maybe between the within generation plasticity to the sort of fixed effects that we see in bombifrons, that's also basically a buying time idea. So again, you're seeing them all come together there. So again, thank you for asking that, because I want to be clear. Talking exclusive pathways to innovation or diversification. Possibly, although people have also argued that plasticity could be really important in large populations, because you have many, many more. If all the individuals in that population are experiencing the same shift in the environment, then you've got a larger pool of heritable variation that can be exposed to selection. So that's one of the ideas that, for instance, Mary Jane Westergaard has argued, is that large populations might actually be more prone to undergoing these kinds of evolutionary shifts because of that effect. But you could argue your way as well. Yeah. Huh?
All right. Thank you all for attending this seminar. Let's give David another hand and thank him for coming out.